जी, बिस्मिल्लाहिर्रहमानिर्रहीम। Today we will discuss about the lab diagnosis, and there are mainly two approaches of the for the diagnosis of the bacteria diseases. Uh, First one is the bacteriological approach, and second one is the immunological approach. आप लोगों ने तो नहीं करना पहले? Firstly, regarding the bacteriological approach, firstly the patient presenting with the sign and symptom, we will collect the specimen, and the specimen as soon as possible they are sent to the lab, and in the lab the first step is a microscopy of the gram stain, right? Second is the culturing, and next is the identification by means of the staining, and side by side these are performing the antimicrobial sensitivity testing. And bacteriological approach includes the first is the most important is the collection of specimen, and it will collect to avoid the contamination, and then the transport the specimen, and providing essential information to guide the laboratory personnel. And in the lab the first step is the microscopy, and then there is obtaining the culture, pure culture, and then specifically identify the organism by using the different biochemical reactions. First step is the specimen collection. The different specimens they are collected in different diseases. For example, in case of the endocarditis, meningitis, osteomyelitis, or pneumonia, there is a collection of the blood specimen, and mainly the organisms which are isolated, they are Staphylococcus aureus and the Streptococcus pneumoniae. And the gram negative includes the E. coli, Klebsiella, and the Pseudomonas. And the specimen is blood sample is collected by the antisepsis of the skin by the pyogene, and it will remove the alcohol and then transfer the blood in the blood culture bottles which contains the liquid media, and then it is sent to the lab, and it is placed in the incubator, and after third day of inoculation it will inoculate in onto the different blood culture medias, and then observe. And second is the throat culture, they are collected whenever there is a patient presenting with the pharyngitis, and three types of organisms they are isolated. For example, group A hemolytic streptococci, coridae, as well as the candida. And here you can see the different membranes in which the throat swab is used to isolate the specimen. And then when the specimen is sent to the lab, it is inoculated on the blood agar, and gram stain is mainly not used to because the presence of the normal flora, right, and it will give the false positive reactions. And here you can see the collection of the specimen in the throat, and it is inoculated on the medias, that is the blood agar, and you can obtain the growth. And in case of sputum, it is collected in two important diseases, for example the pneumonia or the tuberculosis, right? And in case of pneumonia, culture of the sputum on the blood and chocolate, and in case of TB, tuberculosis, the first is the acid frost staining is done, and then the specimen is inoculated on the special media, that is the low stain Jensen medium, right? Here you can see the algae medium, and blood and the chocolate agar. And in case of wound and abscess culture, most common pathogen they are isolated from the wound. They are for example, the gram-positive bacteria includes the Staphylococcus aureus and Streptococcus pyogenes, and gram-negative, that is the Pseudomonas, E. coli, Proteus, and the Klebsiella. And in the lab, they are inoculated mainly on the blood as well as on the McConkey agar. In case of the spinal fluid culture, and it is done in the case of the meningitis, and the most important organism they are isolated from the meningitis, they are the Neisseria streptococcus pneumoniae as the Haemophilus influenzae, right? Here you can see the procedure by which you can collect the CSF, And in case of stool culture, your stool specimen, they are collected whenever there is a cases of the enterocolitis, and the most important organism that can be isolated Shigella, Salmonella, and the Campylobacter, and they are inoculated on the McConkey as well as on the blood agar. And these here you can see the SS agar, that is a selective media, and there is a McConkey agar. And in case of the urinary tract infection, this specimen is collected is the urine, and the most important organism that can cause the UTI are the E. coli, Proteus, Pseudomonas, and the Staphylococcus saprophyticus. And in case of urine culture, the early morning specimen is preferred because it is a concentrated one, and it is cultured on the medium selective as well as differential medium is the clad, which is a cysteine lactose electrolyte deficient medium. And it is a summary of the steps in the isolation and the identification. First the most important is the direct microscopy, followed by the culturing, and then there is a culturing. Whenever there is a culturing is done, there is a after 24 to 48 hours you will obtain the colonies, and then by the performing the gram or the latent sting you will obtain the bacteriological morphology as well as the arrangement, right? Then you will perform the different types of the biochemical test. If there is a gram positive, you will perform the, here you can see the different methods of streaking, in which you will streak the sample on the different medias, and you will place these plates into the incubator, and after 24 to 48 hours you will obtain the isolated colonies. Here you can see the different isolated colonies, and here you can see the colony morphology on the blood agar. Okay, here you can see the hemolysis. On the left side there is a beta hemolysis, and on the right side on the beta hemolysis, and the left side there is a alpha hemolysis. And then you will see the morphology of bacterial colony. Every organism has a different morphology characteristics, and you will perform the gram staining. Okay, in very different reagents they are used, and the gram positive they will appear purple, and the gram negative they will appear the pink colored. And you can see there is a different presentation in which the purple color there is gram positive, and the pink color there is a gram negative organism. These are the gram negative diplococci. And you will perform the different biochemical tests. In case of gram positive cocci, you will perform the catalase coagulase. In case of gram negative cocci, you will perform the oxidase and the sugar fermentation test. You can see the catalase as well as the coagulase test. Here you can see the oxidase test. Right, next the gram negative bacilli, you will perform the different types of the biochemical tests like the indole, citrate, urease, motility, TSI, as well as the oxidase test. And lastly, there is an antibiotic sensitivity testing, which will determine which organism is sensitive against this and which organism is resistant against this antibiotic. Here you can see the zone of clearance around the antibiotic. These are different antibiotics, these are placed, right? And here you can see the zone of clearance, which will determine there is a this drug is highly sensitive against this organism, so you will prefer this drug, right? And it is highly sensitive, and it is a low sensitive, and these are the resistant one. And next approach is the immunological method, and immunological methods, they are done either the detect the presence of the antigen, or there is the presence of the antibody. So there are different methodologies. For example, the identification of an organism with a known antiserum, or the identification of serum antibody with a known antigen, and the nucleic acid base test. First one is the identification of an organism with a known antiserum, that will include the capsular swelling reaction, slide agglutination test, latex agglutination, counter immunolactophirosis, ELISA, and the fluorescent antibody test, in which there is a known antigen, and there is a we will detect the antibodies. And in case of identification of serum antibody with a known antigen, that will include the slide or tube agglutination test or the closed agglutinin test. And nucleic acid based assay, that includes a PCR or other amplifying test. Here you can see the capsule swelling reaction. In the presence of homologous antibody, capsule will swell, and it will detect the mainly the capsulated organism like the Neisseria meningitis, Streptococcus pneumoniae as a Haemophilus influenza. Next is the site agglutination test. In the presence of the homologous antigen and antibody, there is a agglutination occur, okay, which will indicate the positive test. Right here you can see the positive and the negative test. Next is the latex agglutination test. In the latex beads, they are coated with a specific antibody and they will fluoresce under the fluorescent microscopy. And counter-current electrophoresis, in which the antigen antibody, they will move towards each other by applying the electric field. Here you can see there is a presbyterian lines, they are formed. In case of ELISA, enzyme-linked immunosorbent assay, in which the antigen we detect the antigen antibodies by adding the enzyme, which is enzyme labeled, or then we will add the substrate. It will fluoresce under the microscope and give the colored products. Next, fluorescent antibody, in which the antibody they are tagged with the fluorochrome or fluorescent dial, and they will fluoresce under the fluorescent microscope. Then there is a identification of the serum antibody with a known antigen. First method is the tube agglutination, right, in which we can detect there is the formation of the clot. And next are the nucleic acid based assay, in which there is a three most important steps. First one is the denaturation, in which the DNA are the denatured double stranded DNA, they are the single strand, and the temperature of this step is 95 degrees centigrade. Second step is the annealing, in which they are the primers they are added, and the temperature is low down, that is the 50 to 55 degrees centigrade. And then the third step is extension, and the DNA polymerase enzyme is added in this step, and in which the temperature is the 72 degrees centigrade, because this enzyme works at this temperature. Right. DNA probes, in which the isolate the DNA from the body fluid sample, and then you will denature the DNA sample and combine with the DNA probes, and probes are complementary to the gene of interest, and they label with the molecular beacon. A DNA probe will bind to the gene of interest if it is present in the DNA sample. Alright, this is all about the lab diagnosis, mainly two approaches, bacteriological and the immunological. Bacteriological approach, your first important step is the collection of specimen, then you will send to the lab, and then first step is the direct microscopy and followed by culturing, and then you will perform the again you will perform the gram staining, and then specifically identify the bacteria on the basis of the species identification, on the basis of the performing the biochemical test. This is all about bacterial lab diagnosis of the bacterial diseases. Thank you so much. जी